Hello, and welcome to today's session on object and face perception. Today we'll be putting together some of the information that we had in prior lectures when we dealt with lower level visual issues. When we talk about lower level visual issues, we're speaking of issues relating to, for example, feature perception. And we've talked about how we perceive forms, we've talked about how we perceive colors and orientations and so forth. Today we're going to put some of those together and try to understand some visual processes that occur later in the visual pathway. And this might be relating to issues of object recognition and a very special case of object recognition called face perception. Some people have argued that face recognition is actually a highly specialized version of object recognition, and we'll see what the debate is about that and some evidence working for it and some evidence working against it. So the issues today specifically will be, are faces special objects? And then later on we'll get into yet another type of high-level vision called visual imagery. So we'll begin by addressing this debate. Are faces special objects a special case of form processing? Okay, so what we can say is we are able to recognize faces, particularly important faces, despite considerable changes in lighting and in size, and we'll take each of those separately. So as is true for most object recognition, we can recognize faces despite the fact that we might have increasing or decreasing levels of illumination. That is, we can change the illumination hitting a face, and yet your capacity to recognize that face as a particular face is largely unaffected by these lighting changes. It doesn't vary across these lighting change levels, and so it's said to be invariant to lighting changes. Similarly, it's the case that we can say that most facial recognition is invariant to size changes. So there's a level of size invariance. As an example, I could walk into the camera right now, and if I were to do that, my face would expand onto your retinas, and then if I were to move back, it would contract on your retinas. All the while, you would still recognize my face as my face, because you know me through prior videos, and so we can say that we have some kind of size invariance, and we're really quite robust to these kinds of manipulations of size and manipulations of lighting. Just to remind you about a prior lecture, very recently we've discussed Fourier Analyses, and you might recall FAPO as an important acronym there. FAPO is frequency, amplitude, phase, and orientation, FAPO. And so when we're talking about this size invariance for object recognition in general, or for facial recognition in particular, we're talking about some kind of spatial frequency invariance, because spatial frequency is related to size. That said, face recognition is important for information processing relating to mood, and we'll have some conversation in just a moment about how it is that we can use something like a FAPO to extract information about the mood or the emotion that somebody is displaying on their face. Despite all this invariance, we can say that face recognition is disrupted by reversing the contrast polarity from light to dark and vice versa. So you might remember from our FAPO conversation that we talked about phase, or the relative position of a stimulus. And just to remind you of what that slide looked like, let me call that up. Here's the slide from the other day. And so here we have our dark light pattern, dark light. And this stimulus is identical to that in all ways except for phase. If we move this stimulus through 180 degrees of phase, we now have light, dark, as opposed to dark, light. So these two differ only in phase. We can do that also with photographs using Photoshop. We can take a photograph of my face, and the relatively light areas, for example, like the whites of my eyes, we can turn those dark. So we have a light for dark swap, and the darker areas, for example, the darkness of my hair, might become very light in this photographic negative, and this would be an example of a reversed luminance polarity, as we talk about over here. Okay? So face recognition can be disrupted by reversing the contrast polarity, light versus dark. And what we'll do in class is we'll have a couple of demos on this. We'll show you some pictures of famous faces. These are folks you could easily recognize. But when we swipe the dark areas out and swipe in some light areas, you'll see that all of a sudden it's fairly difficult to recognize exactly who that is. And what this tells us is that our face recognition capacity might have some size invariance, as we saw a moment ago. It might be that our facial recognition capacity is spatial frequency invariant, but it is not phase invariant. That is, it depends on phase. It's a very important component of our face recognition capacity, and it might harken back to the activity of some of these simple cells that we learned about the other day. These were the cells in our visual cortex whose firing rate was driven in part by how the light was distributed. There might be on-center cells or off-center cells, and perhaps it's the activity of those kinds of cells that would be playing a very big role in facial recognition and gives us this dependence on the phase of the stimulus. Okay. Although our ability to recognize faces and expressions does not depend on scale, that is, it didn't depend on spatial frequency, relatively high spatial frequency, or small faces, low spatial frequency, or large faces, it does depend on orientation. And we'll show you a second example in class, we won't do these here, of faces that have very peculiar expressions on them. And we first show you these faces in an upside down orientation. So instead of my face being upright like this, we rotate the face now through 180 degrees of orientation as opposed to 180 degrees of phase. And we are unable to detect what kind of expression there might be on that face. Then when we rotate it back until it's upright orientation, and we can readily extract that there's an unusual facial expression of some variety, and sometimes these demonstrations are quite striking. So we'll do that in class also to make the point that our face recognition capacity might be spatial frequency invariant, but it is not orientationally invariant, nor is it phase invariant. Okay. So something else that is rather striking about our capacity to perceive faces under considerable lighting variations and size variations is that we can also extract this very relevant social information that relates to emotion. And we could say that there are typically 6 emotions that are recognizable in faces, and this kind of information comes to us even by people who study cross-cultural psychology. They might have asked questions about how general are the facial expressions that all humans can recognize. There might be some, you might imagine, that are facial expressions that are unique to a particular society, just like there are unique words in specific languages, but there might be some human universals with respect to face recognition. And what the researchers have come to is currently it's believed that there are these 6 typical emotions that everybody can recognize. And they are sadness, disgust, surprise, happiness, anger, and fear. And what we'll show you in our third demo for the day is this demonstration in which you've got faces of varying emotion, and we'll ask people to jot down which of these 6 emotions they think is being exhibited in these different demos, and we'll see if we get some inter-rater reliability. Here's a case where we might not have a type A experiment where there are correct and incorrect answers, and yet somehow there's always a consensus or a near consensus on which of the 6 types of emotions had been expressed in that particular case. It's a fun demo to do in class. Okay. We can also talk about how face recognition breaks down for other reasons. So far today we've mentioned that face recognition can break down when we have a reversal of phase in the Fourier domain. That is, we swap out the light areas and we swap in the dark areas. It breaks down in that case. It also breaks down when we change the orientation by 180 degrees. There are other conditions, though, where face recognition breaks down entirely, and this particular condition has a clinical name called prosopagnosia. Can we all say that together? Prosopagnosia. There you go. Okay, so prosopagnosia is a condition in which a patient knows that a face is a face, but can't recognize whose face it is. So just to give you an example, this person would know that a picture of a face is in fact a face and not a hand, not an elbow, not a computer desktop. It can tell that a face is a face, but it doesn't have the capacity to identify a particular face. Yeah? The inferotemporal brain region is almost always damaged in these patients with prosopagnosia, and we'll just remind ourselves a little bit about where the inferotemporal cortex is. So the notion here is we have infero and temporal, and you remember from Intro to Psychology that we have 4 lobes of the brain, and the lobes over here would be our temporal lobes. So if we think about the inferior portions of our temporal lobes, that would be the inferotemporal cortex. It's sometimes simply known as area IT. To give you just a little bit more neural navigation so that you have this for future lectures, we sometimes talk about regions of the brain that are positioned in a relatively inferior manner, that's down, or superior manner, that's up. And correspondingly, we might have these kinds of linguistic terms. We might talk about a ventral location, which would be down, and a dorsal location would be up. To help you remember those, you can think about the dorsal fin of a shark. We all are familiar with that terminology, or many of us are anyway, that there's a dorsal fin, and that's toward the top, so that will help us with our neural navigation. Back over here, we're talking about the lower regions of the temporal lobe as being relevant to this condition of prosopagnosia. Now here's something that's intriguing in the prosopagnosia literature, and that is even though these folks can't identify faces, they can offer some kind of autonomic nervous system response to familiar faces. So let's take an example. Let's say that we show a patient with prosopagnosia a picture of their mother. The patient might say something like, "I don't know who that is." Their declarative response is one of ignorance. They don't know who that is. But what's interesting is you can make an autonomic nervous system measure and show that there actually is, in some way, a recognition going on. For example, you might have a galvanic skin sensor attached to the person's fingers, and this measures micro levels of sweat. And it might be the case that this person will begin sweating, as revealed by the galvanic skin response, in response to very familiar faces that this person should know, family members, for example, but you don't get that galvanic skin response to persons who are not known to this prosopagnosic patient. So there's a sense in which the nervous system actually is responding to the familiar faces, but the declarative knowledge and the declarative statements would suggest otherwise. Just to help us review a little bit, we'll harken back to what we learned in Intro to Psych about the autonomic nervous system. You might recall that it's one of two branches of the peripheral nervous system. We have the somatic system and the autonomic nervous system. What I'd like you to do is stop the video in just a moment and see if you can recall from Intro to Psych what the two subdivisions were within the autonomic nervous system itself. We typically subdivide that into two systems, and just to refresh your memory, you might stop the video now and see if you can recall those autonomic nervous system divisions. Okay, if you're back, we'll go on. And we'll return to this argument about whether face recognition is really a very special case of object recognition, or whether maybe face recognition is a little bit overrated and isn't something very special. So let's take a look at the arguments that are made on this issue. One argument is that faces are in fact special, and they're special because of two unique characteristics. One is behavioral, and one is physiological. Let's take a look at the behavioral case first. Most people, for example, can classify subcategories of faces, that is, they can pull out individual faces, as quickly as they can get the broad category of face. So in these experiments, what might happen behaviorally is the following. We would show on a computer screen briefly flashed images of faces and non-faces. And we'd get from every participant their reaction time on classifying a particular object as a face or a non-face. And we could measure how many milliseconds elapse for that correct performance. Okay? And this would be a type A kind of experiment, because in a type A experiment, we have correct or incorrect responses. Subsequently, we can have those same folks take a look at a display of faces, one at a time, and we can ask them to maybe identify who that is, and come up with a name like Bill Clinton or Barack Obama or something along these lines. And people can typically identify the names of individuals as quickly as they can identify that it in fact is a face. So this subordinate level classification of particular faces with particular names occurs at the same reaction time level as does the distinction between a face and a non-face. So a very precise level of responding. And this might be relatively unique to faces, or at least that is the claim of people who want to argue that face recognition is actually a highly specialized form of object recognition. Second, there seems to be a well-localized region of the brain, that's area IT, the Infra-temporal cortex, that's dedicated to face recognition. So some folks have argued that we don't typically get the same kinds of IT responses for other types of object recognition, but we clearly get strong IT responses for face recognition. So these two lines of evidence have led some people to believe that face recognition really is a very important and specialized type of object recognition. And that might make sense from an evolutionary standpoint. You can imagine that any species would benefit by being able to recognize not only conspecifics, that is, members of the same species, but also particular members of the same species, people who might be friends, for example, could be especially important to pull out of a crowd if we're in a survival-related situation from an evolutionary perspective. So it might make sense that we have this fast behavioral response and a dedicated neural circuit. Some researchers, though, are a little bit less persuaded by those two lines of research. And they will say that, well, maybe faces really aren't so special, and they argue that instead, non-face stimuli can, under the right circumstances, exhibit these two characteristics: the behavioral characteristic of subordinate classification and the physiological characteristic of IT responding. And to make this point, some researchers, among them a fellow by the name of Dr. Michael Tarr, have created a artificial set of stimuli that he calls greebles. So we're going to meet the greebles in this slide, and greebles look like this. They're rather unique looking stimuli, and that's done for a very precise reason. Dr. Tarr wanted to create stimuli that you had basically no familiarity with. These are stimuli that you hadn't seen before. He created them in his computer. He also creates a whole language around the greebles. He gives every greeble family a unique pronounceable name, but a word that you haven't heard before. Each individual greeble also has a name. Again, it's pronounceable, but it's unique and a name that you wouldn't have heard before. The greebles, like real people and other animals, have sexes, but the sexes are not male and female. The sexes are plock and glip. And again, he uses this terminology because he wants to get away from any associations that you might have between your language and maybe, say, faces, for example. One of the researchers that you learned about in Intro to Psych was a fellow by the name of Ebbinghaus, who was an early researcher in experimental psychology who specialized in memory. And what Ebbinghaus had done was used pronounceable non-words, gibberish words that were pronounceable, and he would test his own memory on this, and he was the one who found the very famous serial position effect, where we remember with great accuracy words that are early in a sequence of words, and words that are very late, the so-called primacy and recency effects, but we lose the words in the middle. And he had used this assemblage or ensemble of pronounceable non-words. That's what Dr. Tarr is using here in an attempt to break any association that you might have with language and faces. So he has these two different sexes, and he has different protrusions that go not by words like ears and nose and chin, but rather he calls this upper protrusion a quiff. Quiff is how that's pronounced. He has boges coming out to the side. The lower protrusion is called a dumph. So people will quickly learn in a greeble sex test that the glips have upward protrusions and the plocks have downward protrusions. So novices are actually quite good at telling, just based on that information, which kind of greeble they're looking at. Are they looking at a glip or are they looking at a plock? So here they have some kind of information about sex, and yet they don't have information about a particular kind of greeble. They don't have subordinate level classification. Okay, but with several thousand trials of practice, that is, Dr. Tarr would bring people in and try to practice identifying a particular greeble and discriminating that greeble from another one and naming the greebles, just like you and I get lots of practice in naming our loved ones as we're growing up. He gives his participants thousands of trials of practice in naming individual greebles. And after a certain amount of practice, typically a person can make subordinate level classification. That is, they can discriminate greeble from non-greeble as quickly as they can discriminate and name one greeble relative to another greeble. So it's really a practice effect. Maybe there was nothing special about faces, according to Mike Tarr's analysis. Also, the more expertise a person has in this subordinate level classification, the higher the level of IT comes in MRI studies. So you might remember that in MRI studies we track something called the BOLD signal, and BOLD is BOLD, and that stands for blood oxygen level dependence. We typically plot that on the y-axis, and then on the x-axis, in this case, we might have something like the number of practice trials that the person has had in classifying greebles. And as the number of practice trials increases and performance gets better, typically the BOLD signal, as measured by MRI, in area IT, or the infratemporal cortex, goes up. So now we have behaviorally this classification for clearly images that are not faces, the subordinate level classification for images that are not faces, and we also have this IT responding as measured by MRI. So people like Mike Tarr would say, well, maybe faces aren't so special after all. An interesting debate for sure. Okay, we'll now move on to another type of high-level vision called visual imagery. And we'll first begin by defining visual imagery, as we have at the top of your slide. Visual imagery is the process of internally experiencing vision in the absence of retinal stimulation. So far this semester, we've spent a lot of time talking about the properties of light. We talked about sensory transduction and how that physical stimulation of electromagnetic energy is converted by way of a cis-trans isomerization into a biochemical process that results in neurons firing and eventually into our psychological experiences of light. But we also can have very salient subjective experiences of vision that don't involve any kind of retinal stimulation. As an example, I always ask my students to do this, and please play along while you're listening to this video. Please, right now, let the video continue to run, but close your eyes. And take this journey with me. I'll ask you to imagine that you are going back to the home in which you were raised. Some of you may have had a few different homes because you traveled or moved. Just pick one of the homes that you're most familiar with, and imagine that you're sitting in your kitchen of this home. We might ask you to see if you can get a strong visual image while sitting in this kitchen, and then we'll ask you to imaginarily get up from this visual kitchen and move around a little bit and go over toward the sink and see if you can recover what color the sink is. And then move over toward the refrigerator, wherever that might be, and see if you can recover the color of the refrigerator. Okay, you can open your eyes now. Most people can do that quite well. It's a type of visual memory that we call visual imagery. Obviously, because we've transported you in this imaginary scenario back to your home kitchen, there was no light from that kitchen landing on your retina, and yet you had a very salient psychological experience of being in that kitchen, and you could see colors, the color of the sink, the color of the refrigerator, and so forth. This is a simple example of visual imagery. Now, some questions arise about how that occurs physiologically. How much anatomical overlap is there between visual imagery, which we just did, and stimulus-driven vision, which is what we've been talking about so far this semester? Again, stimulus-driven vision is something that we would call lower-level vision. It starts out very early in the visual pathway at the point of phototransduction. Visual imagery happens much later. It's believed to be an entirely cortical process. So we can ask questions about how much overlap there might be between these two different kinds of vision, even at a relatively early cortical level, like maybe area V1 that we've talked about before. So some fMRI work has shown that area V1 is active during visual imagery, and you and I already know from prior lectures that area V1 plays a very important role in stimulus-driven vision. Okay. So V1 seems to be playing a role in both this low-level vision driven by the stimulus or this higher-level imaginary vision. Okay. What we'll do in class is now a fourth demo. I won't do this here, but I will show you a demo in class of the kind of experiment that a fellow named Dr. Steve Koslin at Harvard University has done to show that area V1 becomes active when you're engaging in visual imagery and you're lying in an MRI machine. And we're measuring one more time the bold signal as we're going through different kinds of exercises in this visual imagery experiment. In this particular case, we'll be measuring the bold response in area V1 as opposed to the infra-temporal cortex. And he finds that there actually is considerable activity there. So we'll do that demo in class. What we can say now here on the video is this, that Dr. Koslin was able to use, again, a very creative type A experiment to investigate visual imagery. He posed to his participants a series of questions that had factually correct or incorrect answers, presumably questions that couldn't be answered unless you engaged in visual imagery. But yet these questions had externally verifiable answers. So this very subjective experience of visual imagery was actually diagnosed and examined very carefully by Dr. Koslin using a very clever and creative kind of experiment. And it takes the form of a type A experiment. Now what he was doing was essentially eavesdropping on brain activity by using MRI. Some researchers find this to be a persuasive form of evidence that we have overlap between the anatomy and physiology responsible for visual imagery and stimulus-driven vision. Others would say, though, that it might just be that when you're passively eavesdropping on the brain using MRI, you have some correlates, but you don't really have a cause. And there's a difference between having a correlation and having a causal relationship, as we learn even in Intro to Psych. So there are some other ways of getting at whether it's the case that area V1 plays an active role in visual imagery, just as it does in raw visual perception. And what they do to test this out is they introduce a technique that we've not yet talked about so far this semester. And this technique is a technique called Transcranial Magnetic Stimulation, or TMS. And I'll show you a picture of a TMS device in just a moment, but the idea is they apply a magnetic pulse to area V1, and they do this when the person is looking at stimuli that are landing on the retina, and they do this also when the person is engaged in visual imagery. And what they find is a very similar outcome in those two cases. That is, TMS to area V1 can slow the response that a participant might make by about 200 milliseconds. And this is a slowing that occurs when TMS is applied to area V1, but perhaps not when TMS is applied to maybe Broca's area, which is believed to be unrelated to vision. So what's neat about this is TMS allows us to actually manipulate the state of the brain rather than just eavesdrop on the state of the brain. So we're getting something closer to a causal relationship by using this manipulation, whereas back here in MRI, we're getting something more akin to a correlation. Just to complete that thought, here we have some diagrams relating the pictures from a TMS experiment, and here is the TMS device. This item right over here is a capacitor, which holds a charge, and it sends out an electrical charge to this device, which will put out a magnetic pulse. And this device will be held maybe against the area V1 or against Broca's area, or wherever the case might be, and we put out a very brief but very intense magnetic pulse. Some of you might remember from physics classes that magnetic pulses are measured in Tesla, and you can systematically manipulate the magnitude of this magnetic pulse by changing the Tesla level. And when you do put out this magnetic pulse, you very momentarily change the electrical state in some region of the brain. And this is related to a notion from physics called Faraday's principle, which talks about how when there is electricity flowing in one direction, there is a magnetic field flowing in the opposite direction, or in a perpendicular direction. Some of us might remember the so-called right-hand rule from physics that speaks about the relationship between electricity and magnetism. Well, in TMS, we take advantage of that, and what we do is apply this magnetic pulse to different regions of the brain and selectively disrupt momentarily the activity of the brain. On these axes, we have just 1 millisecond of time, and you can see that within 1 millisecond, we get a very strong change in the magnetic field and a corresponding change in the electrical field, probably just on the order of 1 millisecond or so. And yet that alone is enough to momentarily disrupt a behavior kind of like the visual imagery behaviors or the visual recognition behaviors that Steve Kosslyn might test in his experiment. So what we'll do is have you, as always, jot down what you thought was particularly clear about this video presentation, and also what was less clear, and you can bring those comments to class. See you in class.